There is an organ in the human body that for centuries was considered dispensable, an organ that medieval physicians blamed for melancholy, that 21st century surgeons removed without much concern, and that until a few decades ago was treated as a biological accessory. An organ that nevertheless works every second of your life, filtering your blood, destroying millions of defective cells, storing emergency reserves, and coordinating immune defenses with a sophistication we still don't fully understand. That organ is called the spleen, and its history is, in reality, the story of how long it took us humans to learn to truly see what's inside us. The spleen lives hidden. It is located in the upper left quadrant of the abdomen, just below the diaphragm, protected by the lower ribs, as if the body knew it couldn't afford to lose it. In a healthy adult, it measures between 9 and 13 centimeters in length and weighs approximately 150 grams, although this figure can vary enormously depending on health, hydration, and even the time of day. It has the vaguely oval shape of a large bean with a convex surface that rests against the diaphragm and a concave inner surface where the blood vessels that feed and drain it converge. This vascular entrance, known as the splenic hilum, is the gateway through which everything passes. Blood in, blood out, signals arriving, and waste products leaving. From the outside, its texture is smooth, almost fragile, and that fragility is not a misleading impression. The spleen is one of the organs most vulnerable to trauma. A hard blow to the left abdomen can rupture it, and when that happens, the result is internal bleeding that can be life-threatening within minutes. Emergency surgeons know this well. For decades, the standard response to a ruptured spleen was splenectomy, complete removal. And patients survived. They lived for decades without it. What took us a while to understand was that surviving without a spleen is not the same as living completely protected. To understand why, we have to go inside the organ. We must abandon the visible scale and descend to the microscopic architecture that makes the spleen completely different from any other structure in the body. The spleen has no ducts, it doesn't produce digestive juices, and it doesn't secrete hormones into the bloodstream in the way the pancreas or adrenal glands do. What the spleen does is more difficult to describe in a single word. It filters. But that word falls short of capturing the true complexity of the process. The spleen is not a passive filter like those used in industry. It is an active, intelligent, ever-changing filter that makes moment-to-moment decisions about what remains in the blood and what must be eliminated. Blood enters the vessel through the splenic artery, which branches into increasingly smaller arteries until it becomes arterioles that crisscross the splenic tissue in all directions. And here's where something extraordinary begins. These arterioles don't drain directly into veins. In much of the vessel, the blood flows into open spaces, into spongy tissue cavities called sinusoids, where it comes into direct contact with specialized cells. This architecture, called open circulation, is unique in the human cardiovascular system. In almost every other part of the body, blood travels within closed vessels. In the spleen, blood is released into a porous labyrinth where each red blood cell, each platelet, each immune cell must squeeze through narrow spaces, almost as if passing through a biological sieve. This sieve has its own design. The splenic sinusoids are formed by endothelial cells that create walls with slits only 2 to 3 micrometers wide. A healthy red blood cell measures around 8 microns in diameter, but it is extraordinarily flexible. It can deform, elongate, and compress itself to squeeze through spaces smaller than itself. An old, damaged, or defective red blood cell loses this flexibility. Their membranes become rigid, and when they reach the sinusoids of the blood vessel and can no longer pass through, the macrophages that guard that space recognize them, trap them, and destroy them with remarkable efficiency. This process occurs constantly, without pause, throughout life. Each red blood cell has a lifespan of approximately 120 days. When that time is up, it reaches the blood vessel, and there its cycle ends. It is estimated that the blood vessel destroys between 150 and 200 million red blood cells per hour. Per hour, not per day, not per week, but per hour. And the components of those destroyed cells are not lost. The iron contained in the hemoglobin is recovered, sent to the liver, and reused to make new red blood cells. The spleen is, among other things, a continuously operating precision recycling plant. But mechanical filtration is only one of its functions. Alongside it operates something more elaborate, something we only began to understand clearly in the second half of the 20th century. Within the spleen, there are two types of tissue with completely different compositions. The first is called red pulp, and it is where the filtration we just described takes place. It is the territory of macrophages, blood, and sinusoids. But the second type of tissue, white pulp, is something else entirely. White pulp is, in essence, lymphoid tissue. It is an immune territory, and its internal organization reflects this function with an elegance that is hard not to admire. Around the central arterioles of the vessel, a dense sheath of T lymphocytes forms, immune cells specialized in recognizing threats and coordinating responses. Surrounding this sheath are follicles of B lymphocytes, responsible for producing antibodies. And between the white pulp and the red pulp lies a transitional region called the marginal zone, where cells with a special ability are concentrated. They can recognize encapsulated bacteria, microorganisms wrapped in a polysaccharide capsule that makes them resistant to other forms of defense. This marginal zone is the only place in the body where this specific surveillance occurs so efficiently. Here's why splenectomized patients live with an increased risk of certain infections. Streptococcus pneumoniae, Haemophilus influenzae, Neisseria meningitidis, encapsulated bacteria that, in a person with a spleen, are quickly intercepted and destroyed, but in the absence of the organ can spread through the bloodstream before the rest of the immune system has a chance to react. This condition has a clinical name, fulminant post-splenectomy bacterial infection syndrome. It's not common, but when it occurs, it can progress within hours. This is why doctors today try to preserve the spleen whenever possible. And when that's not possible, they routinely vaccinate patients without a spleen against these specific organisms. The spleen also stores something the body might need in an emergency. Within the red pulp lies a reserve of platelets. Not all platelets circulate freely in the blood. Approximately 1/3 of the total are stored in the spleen, ready to be released in the event of bleeding or an acute demand. The spleen can literally contract in response to signals from the autonomic nervous system, expelling this reserve into the bloodstream in seconds. This splenic reflex, well documented in diving animals such as seals and dolphins, has also been observed in humans. Freedivers exhibit a significantly smaller spleen during deep dives, evidence that the organ compresses to optimize oxygen transport under oxygen deprivation conditions. Research in this area continues, and there is still no consensus on the functional extent of this mechanism in humans under everyday conditions. The history of knowledge about the spleen is rife with misinterpretations that persisted for centuries. In Galenic medicine, which dominated medical thought for over 1,000 years, the spleen was considered the source of black bile, one of the 4 humors supposedly regulating health and temperament: melancholy, deep sadness, and pessimism. All of this was attributed to an overactive spleen. The very term splenic, which we use today to refer to the organ, shared a root with an Old English word meaning dark or grumpy humor. During the Renaissance, anatomists began to describe it more precisely, but its function remained a mystery. Vesalius, in the 15th century, drew the vessel with remarkable anatomical accuracy, but he could not explain its purpose. Now Pigi, in the 17th century, identified the corpuscles that now bear his name, the lymphoid nodules of the white pulp. But without a conceptual framework for the immune system, it was impossible to understand what he had seen. The path to true understanding required waiting for the development of modern immunology. It was in the 20th century, with the development of electron microscopy, cell markers, and experimental animal models, that the image we have today of the spleen as a first-rate immunological organ began to take shape. Not an obsolete appendage, but an active and irreplaceable component of the body's biological surveillance. There are still aspects that research is exploring: the relationship between the spleen and the gut microbiome axis, the possible splenic influence on chronic inflammatory states, the role of dendritic cells residing in its white pulp, and the precise dynamics of the marginal zone in complex immune responses. These are areas where science is advancing, but where definitive answers are still lacking. In recent years, research on the spleen has taken an unexpected turn. Studies in animal models have suggested that the spleen may act as a reservoir for monocytes, immune system cells that respond to inflammation and tissue damage. In the event of a myocardial infarction, for example, the spleen has been observed to release large numbers of monocytes that migrate to the damaged heart and participate in the repair processes. Whether and to what extent this mechanism operates similarly in humans is something researchers are still evaluating. What this line of research suggests is that the spleen may have an influence on organ recovery that goes beyond what we had previously assumed. There's no way to finish talking about the spleen without pausing for a moment to consider what its history reveals about human biology in general. For centuries, we assumed we could do without it. We treated it as redundant, as if the body had made the mistake of including something unnecessary in its design. And that assumption consistently proved incorrect. The spleen turned out to be essential for defense against certain pathogens, a regulator of platelets, a filter of aged red blood cells, a coordinator of immune responses, an emergency reservoir, and a potential participant in tissue repair. This accumulation of functions, all occurring silently, invisibly, and continuously, is characteristic of almost every system in the human body. Biology rarely produces structures with a single purpose. Redundancy, multifunctionality, integration between seemingly separate systems. These are the principles that govern the architecture of the organism, and the spleen embodies them with a clarity that is instructive, not only for understanding this specific organ, but also for learning to look at the human body with greater humility. Inside you, right now, there is an organ working silently. It is destroying millions of old cells and releasing their components to build new ones. It's monitoring the blood flowing through it. Searching for bacteria it recognizes through molecular signals that humans are only just beginning to decipher. It's storing platelets in reserve, just in case you need them. It's making filtering decisions that no one orders it to, following a logic embedded in its architecture since before you were born. You don't know it's there, but it doesn't need you to know to do its job. The spleen is proof that the body holds more intelligence than we are capable of seeing. For centuries we ignored it, underestimated it, removed it without remorse. And every time science revisited it with better tools, it found something new. That should tell us something about everything else we think we know. If this journey through the hidden architecture of the human body gave you that feeling of seeing something for the first time, leave a like. It's the signal that tells the channel it's worth continuing. And if you want a different organ, system, or biological mechanism to be revealed each week with the same level of depth, subscribe to Revealed Anatomy. The human body has many more secrets to uncover.